Yes, level biology 9700, paper 2, variant 2, October November 2025. And in this video, we will discuss question number 3. And question number 3 is the gene LCT codes for the enzyme lactase. In babies, lactase synthesis is necessary for digesting lactose, the sugar found in milk. Another gene, MCM6, has introns that has a regulatory role in the expression of gene LCT. So gene MCM6 codes for a protein that has no involvement in lactase synthesis. As children get older, the MC-M6 introns are responsible for a decrease in lactase synthesis. This decrease in lactase synthesis is known as lactose non-persistence. LCT and MC-M6 are located on the same chromosome in humans, suggest differences between gene LCT and gene MC-M6 other than their locations in different positions on the same chromosomes. So they are different genes, so they will naturally have different primary sequences and different roles in the cell. So we have to suggest the differences which are based on the information provided and general biological principles. So first of all, we will go with the first difference which I would like to write is the structural difference. So each gene has a different base sequence which allow them to code for different polypeptide. And second, if I go with the MCM6 gene contains introns that regulate the expression of the LCT gene, whereas LCT itself does not have a regulatory role over MCM6. And then it is highly likely that they have a different total number of nucleotides or base pairs. So they will have a different number or different arrangements of introns and exons. So in this way, we can write these differences over here. So I am going to write all these. First of all, each has a different base sequence. Which allows them to code for different polypeptides. They have a different total number of nucleotides the MCM M6 gene contains introns that regulate the expression expression of the LCT gene, whereas LCT itself does not have a regulatory role. They have a different number or different arrangements of introns and exons. Now the next part of this question is B part, which is with reference to the process of lactase synthesis, explain the relationship between a transcribed strand and a primary transcript and a messenger RNA. So here, first of all, the transcribed strand and primary transcript. So here it is important to distinguish between that happens during transcription and the subsequent post-transcriptional modification. So first of all, the transcribed strand versus primary transcript. So the transcribed strand known as the template strand or which is the specific DNA strand that RNA polymerase reads to build the RNA molecule. So the primary transcript is the direct product of the transcription. Its nucleotide sequence is complementary to the transcribed DNA strand. So because of the base pairing rules, the primary transcript will be a copy of coding DNA strand, but with uracil replacing the thymine. Yes, here in RNA, there is uracil instead of thymine. So this is the relationship and between both of these. So second one is the primary transcript and the messenger RNA. So the primary transcript uh, isn't ready for translation immediately after it is formed. So messenger RNA is the final mature molecule that remains after splicing is complete. So it consists only of the exons that will actually be translated into the polypeptide chains for the lactase enzyme. So if we talk about the relationship between them, the primary transcript contains both exons and introns. So through a process called splicing, the introns are removed and the exons are joined together. So this is of only 2 marks. So directly we have to write the first one transcribed, the transcribed strand is also called as template strand. Template strand which is the specific DNA strand that RNA polymerase reads to build RNA molecules. The primary transcript is the direct product of transcription. So its nucleotides sequence is complementary to the transcribed strand. And now the messenger RNA. Messenger RNA is the final mature molecule that remain after splicing is complete. It contains only of the exons. Which will be translated into polypeptide. Now, the next question, part of this question is C part. A mutation is a regulatory intron of MCM6 allows lactase synthesis to continue. This is known as lactase persistence. In this mutation, the number of nucleotides in the introns remains the same, but one of the nucleotides is different to the original nucleotides. So state the type of mutation that is the cause of lactase persistence. So here It specifies that the number of nucleotides in the intron remains the same, but one of the nucleotides is different to the original. So this indicates that one base has been swapped or for another without adding or removing any genetic material. So when we identify gene mutations, then we can see that this is a type of substitution. The type of mutation is a substitution. Because the one base has been swapped for another without adding or removing any genetic material. So this base count stays the same, only one base changes. So total base count increase can cause a frame shift. So this is the specific for substitution. Okay, now moving towards the next part of this question. Figure 3.1 summarizes the reaction catalyzed by lactase. So draw the ring structure of alpha glucose in the box provided to complete figure 3.1. Here lactose will be broken down. It will be hydrolyzed by adding water into it in the presence of lactase enzyme. So there will be the formation of galactose and alpha glucose. So I would like to draw the alpha glucose right over here. Now CH2OH and H, then there will be oxygen over here and hydrogen OH. So it will be this glycosidic bond will break down, due to which there will be the formation of galactose and alpha glucose. Now E part, some people with lactase non-persistence may be lactose intolerant. They may have symptoms such as abdominal pain. If their diet contains lactose, lactase supplements, tablets can be taken before milk or milk-based products are consumed to avoid symptoms of lactose intolerance. A student compared the activity of two different concentrations of a lactase supplement using an artificial substrate ONPG instead of lactose. A solution of ONPG is colorless, but the hydrolysis of ONPG by lactase releases a colored product. The student planned to follow the progress of the reaction for such of each concentration of lactase using a colorimeter. Explain why the student choose to use a colorimeter to follow the progress of the reaction for each concentration of lactase. So explain why the student choose so to use a colorimeter. So here we can explain the use of a colorimeter. So we can use the colorimeter. The hydrolysis of ONPG by lactase releases a colored product. This color change is what allows a colorimeter to be used. Secondly, a colorimeter measures color intensity. A colorimeter is an instrument that measures how much light a colored solution absorbs. So it measures the absorbance. So deeper the color, the more light is absorbed. So the color intensity is proportional to the product concentration. As the reaction progresses, more of the colored product is produced. So this makes the solution darker in color. By taking readings over time, the student can track the increasing absorbance, which directly responds to the increasing concentration of the colored product being formed. So this is the reason they the student choose to use a colorimeter. So I would like to two of the uses of this colorimeter, two of the reasons in other sense you can see a colorimeter measures color intensity. It is an instrument that measures how much light a colored solution absorbs. The deeper the color, the more light is absorbed. Second point we can write as by taking readings over time, the student can track the increasing absorbance. And third point, we can write that due to a color change when the hydrolysis of ONPG by lactase releases a colored product. This color is what allows a colorimeter to be used. Well, the next part of this question is the lactose in milk can be hydrolyzed using immobilized lactase or using lactase free in solution, free lactase. So this results in milk and milk products that do not contain lactose and so are suitable for lactose intolerant people. Scientists carried out an investigation to compare the activity of lactase immobilized in very small magnetic beads with free lactase at different temperatures and at different pH values. Figure 3.2 shows the activity of immobilized lactase and the activity of the free lactase at 5 different temperatures. Figure 3.3 shows the activity of immobilized lactase and the activity of free lactose at 8 different pH values. So you can see the graph percentage of maximum activity at different temperatures. So immobilized lactase has the curve is on right side and on left side the free lactase curve can be seen. And in figure 3.2 you can see the different pH values, the percentage of maximum activity of immobilized lactase and free lactase. Now the question is one advantage of using magnetic microspheres with immobilized lactase is that they can be easily recovered using an electric field and reused. With reference to figure 3.2 and 3.3, explain why the results indicate that there are other advantages in using immobilized lactase instead of free lactose to produce lactose-free products. So the question is that we have to explain. So we have to if we observe the figure 3.2 and 3.3, then we can have the advantages of using immobilized lactase. So first of all, we can say that the thermal stability of is there at temperature above 40 degrees C. You can see the temperature above 40 degrees C. This is the 40 degrees C, and above the activity of immobilized eggs remains significantly high. You can see this curve is significantly high, and whereas this is on the left side, so it is lower. So for at 45 degrees centigrade, you can see it is 80%. So of its maximum, while free lactase drops, it is 80%, and it it is below this at this temperature at 45 degrees centigrade. So it is lower than this. So the immobilized process to the stabilizes the tertiary structure of the enzyme. So this makes it less susceptible to denaturation by heat, allowing the reaction to be carried out at higher temperatures for fastest reaction rates without losing as much as enzyme function. So on the other hand, if we go with the pH value, then you can see over here in this again, the curve uh, is maximum in both cases. You can see that. The immobilized lactase maintains a higher concentration of its maximum activity across a wider range. You can see that this is the particularly visible in both the acidic range 5.5 and 6.5. So this you can see over here. So and the alkaline range is between, this is the alkaline range and this is the acidic range. So the beads provide a micro environment that protects the enzyme's active site from extreme changes in hydrogen ion concentration. So we can say that is likely, this means the enzyme is less likely to denature or have its active site charge altered when the pH fluctuates, making the problem more robust for industrial milk treatment. You can see in both curves. You can observe it in both. So now we have to write the answer after this explanation by keeping in mind the both of these graphs with pH and temperature. So I would like to write higher activity at high temperature. So this is the first point, is observed immobilized lactase is more resistant to denaturation by heat, showing maximum activity. Than other enzymes. It maintains it maintains higher activity at a wider range of pH values compared to free lactase. So if we go with the temperature, we can see immobilized lactase remains higher than that of free lactase even at 45 degrees centigrade and shows maximum activity 80%, while free lactase drops to about 50%. So it was all about this question. Please like, share, and subscribe my channel. Thanks for watching and wait for next video.